Do you even got a vaccine or eating food from grocery store? And do you even realize that how some apples are not turned to brown, although they're exposed to the air? The question is, how does scientists do this?
Well, to begin with, let us know first what is DNA. DNA is deoxyribonucleic acid, which is made of a long stretch of connected nuclear bases, which is adenine, thymine, guanine, and cytosine. DNA contains genetic information that express our traits.
With DNA technology, Herbert Boyer and Stanley Cohen attempt to manipulate the genotype and phenotype of an organism to make it change from its looks and its function by forming a clone. In biology, clone is a group of individuals that is genetically identical because identical daughter cells are produced during cell replication. So, to form DNA cloning or a clone, scientists cut out one or two fragments from one DNA to insert it in another DNA, which made in vitro. This method called as recombinant DNA technology.
To work out recombinant DNA technology process, we need tools. Okay, first, we need target DNA. Target DNA acts as the sequence of DNA containing of gene of interest from one organism that we can snip it out to combine with another organism. Okay, next, we need restriction enzyme. The enzyme that help to cut and snip the DNA molecules at restriction site and produce restriction fragments. As an example, EcoR1. This enzyme makes staggered cut within the sequence and produce sticky ends, which is single-stranded ends. Another example is SMAR1, produced by Serratia Marcescent. The cut is made between letters C and G and it produces blunt ends. Third, DNA cloning vector. What is DNA cloning vector? DNA cloning vector is DNA molecule that can carry foreign DNA in a host cell and then replicate that. What is the characteristic of DNA cloning vector? First, it's able to accept the foreign DNA in the multiple cloning site. Second, it's able to replicate in host cell. Third, it has selectable genetic mark, which means it has resistance to the antibiotic. For example, ampicillin R. Lastly, we use host cell as a tool. What is host cell? Host cell is bacterial or animal, or it can be plant cell that can serve as a living host for expression of recombinant DNA. This tool has its own characteristic. There has three. Number one is it is able to receive the recombinant DNA. And the second, it is able to maintain the structure of recombinant DNA. And the last one, it is able to amplify it.
So, there are 5 steps in gene cloning. The first one is isolation of DNA. The second one, cut or clip. The third one, insertion. The fourth one is transformation or amplification. And the last one is screening.
First step in cloning a gene is to isolate the DNA from organisms that contain a target gene.
Isolated DNA will be purified and fragmented with restriction enzyme to cut it into fragments known as restriction fragments. After the restriction fragments are formed, then the target gene must be separate from the other restriction fragment by using gel electrophoresis and DNA probe. So, what is the function of gel electrophoresis? The function of gel electrophoresis is to separate the DNA fragment according to their size, while the DNA probe will identify and locate the target gene. As a result, we will get target gene that will be inserted into DNA cloning vector.
Examples of DNA cloning vector that usually used is plasmid. Other than that, plasmid must have LAC-Z, which is LAC-Z contains of restriction site and synthesis of beta-galactosidase. This LAC-Z that contains the restriction site will be cut by using restriction enzyme. So, the next step is
insertion of target gene into a DNA cloning vector. The target gene must insert in DNA cloning vector to form recombinant DNA. So, the exposed base on sticky ends of plasmids will attract to another basis of sticky ends of target DNA to form hydrogen bond, or plasmids that do not carry target gene due to the reannealing of their own sticky ends called non-recombinant plasmid. The DNA fragments then are joined by using enzyme DNA ligase to form covalent bond between nucleotides to produce recombinant plasmid.
Next, transformation and amplification. The recombinant DNA then insert into the bacteria by the transformation process. The bacteria reproduce. The recombinant DNA is also replicated. The last step is screening. Screening helps to identify the bacteria clone that containing the recombinant DNA. And then, the blue-white screening is done.